Okay, so the goal of this study was to better understand how SARS-CoV-2 affects lung epithelial barrier cells and their function. The reason why we chose the lung epithelial cells was because this is our first line of defense against environmental stimuli. And with that being said, we transfected some human lung epithelial cells called Calu-3, and half of them were spiked with the pseudotype VSV SARS-CoV-2, and the other half were spiked with a control. After 2 days, these cells were harvested and we analyzed the epithelial functions and some of their barrier functions as well. What we found was some epithelial barrier dysfunction. Over here, we see some immunofluorescence images. And what this shows is that our transfection was effective. Using transepithelial resistance, we found that transepithelial electrical resistance, we found that we lost cell integrity because the electrical resistance decreased. We used the Fitzy-Dextrin assay to analyze the cellular permeability, and we found a significant increase of that cell permeability, which means we lost that cell-to-cell adhesion in our epithelial cells. And we also used a gene called ZO2, which is associated with tight junction function, and it had a downregulation of gene expression, which means that we've lost that tight junction function. We also analyzed an array of genes associated with inflammatory response. Over here, our 6 genes are shown, and across the board, we found that there was an upregulation. And this makes sense because with those spiked cells, we would see an increase in inflammatory response when that pseudotyped SARS-CoV-2 is present. Moving forward, we want to continue our studies on the gene expression. We want to better understand how Calu-3 cells, pseudotyped infected Calu-3 cells compare to cells from SARS-CoV-2 patients, from influenza A patients, and even patients who don't have that infection as well, so just a control. Thank you.